Brazil is also going through, pandemic, even worse in many instances than what we have here in India. And so I haven't had any visiting people from there. I also work quite a bit with people in US, and I'm just listing some of the main collaborations whose work I'm going to actually talk about today. So I want to give a very brief background. So I always try to give a brief background about where I am from. Well, I already told you guys that I'm actually originally from Baroda, Ahmedabad area, but where I am currently, so I'm in a city which is called Fort Collins, Colorado. For some of you who know the map of United States, Colorado is a state which is sort of, we always consider Colorado as sort of a mountain state or a western state. Because half of our state is mountains and half our state is flat, and we have Rocky Mountains, and I have a couple of pictures about that. The biggest city in Colorado is Denver, which I am sure you may have heard about it. This is where our airport is. This is where I fly. And the university is in a town which is called Fort Collins, which is about 60 miles, maybe about 100 so kilometers from Denver, which is where the university is, which is where I live, and my lab is there. Our city is a very small city. We have about 2 lakh population, 200,000 population. Yeah. It's a very small, it's considered a pretty big city for the state because it's a very, very wide open state. But it's a very small city compared to India. University is the main employer of the university, of the city. But just to give you an idea about some of the pictures that of the city, this is how from the campus it looks like. About 10 minutes you drive and you're in these Rocky Mountains. Rocky Mountains, they don't have a lot of trees; it's just rocks, and they have pine trees. But we get a lot of snow. We get a lot of heat also. Like just last week when I came, our temperature was almost 37 degrees. Whereas here it's not 37, but it's still very hot because it's very humid. For us, it's not very humid. But in summer it's really nice. The university is a very old university. It was founded in 1870 as an agricultural college. This is how all the universities in US were formed, because at that point, agriculture was really important. How to feed the people, how to make sure that people have good quality food. And that's how it kind of started as agriculture. It eventually became agriculture and mechanical, and now it's a state university. We have about 28,000 students, about 1,600 faculty. And this is how our campus looks like. So this is typically in a summer, you would see, a lot of greenery. Whereas in the winter, the same area, you can see we have a lot of snow. We get a lot of snow. In winter. And this past winter was really cold. I mean, our temperature can go all the way to minus 25 degrees C. And as I said, in summer, it can go to plus 37 degrees. So we have this huge change in the temperature, which is there. But we do get really nice autumn season where you can see the color of the trees are also changing. The campus, this is one of the pictures of the campus. You can see this is sort of the central part of the campus. Pre-pandemic, it's a very happening place. This is where everyone kind of, meets and, when you're going from one class to other class. This is our student center, which is, as you can see, there's nobody because the picture was taken during the pandemic. We do have a stadium as well because a lot of universities in US, they have this team, so we have a football team, American football team, which does that. My lab, it's called the Biomaterials Surface Micro Nano Engineering Lab, because we work on surfaces and we try to engineer the surface at a micro and a nano level. And my lab is located in this building, which is a relatively new building. Well, if you think about IIT Gandhinagar, it's much more newer than our building, but this is about 8 years old building. Which basically was a dedicated building for bioengineering research. And actually, the concept is very interesting. We don't have our own individual labs. It's all shared labs. Everyone shares a lab. Everyone has their own equipment and everything, but everyone still shares their lab. And so we have easy access to different equipments and all these things. So this is just a very typical lab that I have. It's just a very standard chemistry materials kind of lab. We also have a cell culture lab where we can do all the microscopy and all those kind of things. And then university has central facilities where we can actually go and do electron microscopy and some high level sort of material characterization. So that's all, sort of a brief about where I'm from and the university and everything. In the next 30, 35 minutes or 40 minutes or so, I'll try to give you sort of a brief overview of some of the work that we have been doing in my lab, which is basically focused on biomaterials. And as you can see, this slide kind of gives you a few examples of some of the things that we have worked in my lab over the years. Materials are very commonly used for biomedical applications. Any kind of a medical device is made up of materials. You do have materials, which are sort of the integral part of medical devices. Now, medical devices can range from anything like catheter, which you're going to put inside the body for a few hours, to all the way up to something like an orthopedic implant. And this orthopedic implant is typically the one which you would actually have it in a body for maybe 10, 12, 15 years. So you do have a lot of these different types of applications where materials are being used. Many times you're using the material by itself, like for example, like a hip implant, you have a titanium stem, but many times you also have combination where you have a polymer metal sort of medical device which is used. This is just an example. A lot of our work is focused on metals and metallic materials, but we also work on polymers as well. Today's talk is going to be mostly focused on metals. I'm going to talk mostly about orthopedic implants, like hip and knee implants, and then I'm going to talk a little bit about stents, which are also basically made from which are basically made from an alloy. It's a nickel titanium alloy from which they're actually made. Now, one of the things which is really critical when a material comes in contact with the body is the surface. So as soon as you take a material, put it in the body, the first thing that comes in contact with biological environment is the surface. And the surface properties are actually going to govern what's going to happen on the material surface. So for example, if your surface is hydrophilic versus hydrophobic, you'll have a very different interaction that's going to go on the surface. If you have a different type of topography, like for example, if you have a porous surface versus a non-porous surface, you're going to see that proteins are going to interact differently, cells are going to interact differently. This cartoon here kind of shows about what are the kind of things that can go wrong on a material surface. If you actually have, certain kind of proteins that would adsorb, that would activate certain kinds of cascades, then you eventually end up into rejection of implants or rejection of materials, and then it could be a lot of problem. So we want to design these material surfaces such that we can control what kind of interactions are going to happen. And that's sort of the holy grail in the field of biomaterials, is how do we make these materials more adaptable, more compatible for a specific type of an application they're going to be used for. And that's what our focus has been. Our focus has been, how do we know that what kind of surfaces we should make? So we go back to biology. And I always tell my students, everyone who works in my lab, that, you need to be, sort of equally versed in biology as well as in engineering. Because if you don't know the biology, you can't solve the problems using engineering approaches. So we have a very engineering approach to think about tissues. We always think about tissues as a material. And when you think about tissue as a material, you can kind of deconstruct tissue into its components. Basically, you have cells and everything which is not the cells. Everything which is the cells, they are actually called, everything which is the cells, they are actually everything which is not the cells, that's actually the extracellular matrix, the ECM. And the ECM is actually, the really important component of the tissue, because it gives tissue the properties. It gives tissue, its elasticity, its rigidity, its hard, soft. It's all because of the ECM. But if you think about it from an engineering perspective, this ECM is a very interesting material, because it's a material in which the cells live, and cells are really happy, and cells are actually able to do what they need to do in the ECM. And combination of cells with ECM is basically forming what the tissue is. But if you start sort of looking at different structural aspects and biochemical aspects, you can see that ECM is actually made up of nanomaterials. It's actually made up of fibers, proteins, small molecules. It's porous. It has fibrous structure. Depending on the tissue, it has different architecture. And so that basically not only gives the biochemical properties, but also gives the structural properties. And cells are used to living into this ECM. So if you start kind of understanding how the ECM is built and how ECM is functioning, you have the ability to kind of figure out what kind of environment the cells need so that they can be as much in a natural environment as they would be. And now if we actually take material surfaces and put on these material surfaces similar type of biochemical as well as topographical cues as that in the ECM, then the cells, when they come in contact with material surfaces, hopefully should have a better integration. That's what our goal is. And so by understanding ECM, we can come up with biomolecular cues and the topographical cues. And we use this sort of principle now with orthopedic applications and cardiovascular, which is the two applications that I'm going to talk about. So the first thing I want to talk about is about orthopedics. Orthopedics is something, orthopedic implants are very commonly used for people, who have joint problems. After a certain age, the doctor would recommend that you replace your knees or hip. Many times people, when they have accidents, they end up getting implants as well. And the number of implants is increasing. This is some statistics I have from US, which shows that by 2030, more than 20% of the people between age, over the age 65 are going to have an implant, are going to have some type of an orthopedic implant. And as the population grows, keeps on aging, these implants we have to make them even better, because right now, when you put these implants, within 10 to 12 or 15 years, they fail. They fail because the bond which is formed between the bone and the material, it breaks and you have loosening of the implant that happens. And many times this loosening of the implant causes pain and inflammation. Many times you have the cup and ball joint, and you are in a hip implant, and it's constantly moving. That cup, which is made up of polymer or sometimes ceramics, you will have wear particles and debris that can go into the gap between the bone and the implant, and that's also going to end up making it making more complicated for these patients to be able to. Keep on, living with those implants, and revision surgeries are going to be needed. The most common material which is used for orthopedic implants is titanium-based alloys. Titanium has been sort of like the two go-to material, but people have made alloys out of titanium because titanium by itself in pure form is not going to work. Here is a cartoon that I have given here, just kind of gives an idea about what I was talking about. So you have this hip implant with a metallic stem that's actually put into the bone. This is a hip implant, and typically you can put a bone cement so that it sticks. And over time, people have moved away from this bone cemented implants, because these cemented implants do have a problem with wear. So they have also moved away from some of these. So titanium is really sort of the key material because it has some really good properties, which actually would help in terms of long-term stability of these implants. You can't use stainless steel because corrosion is a problem. You can't use, some of the other, you can't use aluminum because it doesn't have good, strength. So there are a lot of sort of requirements which are there for orthopedic applications. And it's not just orthopedic, it's also even the dental applications. And you can see here in this cartoon that there are many, many different types of implants which are made from titanium, whether they are dental implants, a lot of craniofacial implants, which you do the reconstruction of the face. They are made from titanium alloys, shoulder implants, spinal implants, knee implants, hip implants, bones, screws, and plates, and a lot of different types of implants are made from titanium-based materials. The reason why, because titanium is biocompatible with orthopedic system. You do have good properties of titanium for this. Right now, the implants which exist in market, they do have a texture. 20, 30 years, 40 years back, 50 years back, all the research which was done basically showed that you can have smooth titanium on implant surface. You do have to have some type of a texture. And typically that texture is at a micro scale, about 200 to 400 microns. And so right now, a lot of the commercially available implants, they would have micro machine patterns, or they would have some plasma treatment. They would have some etching to make that surface rough, because that rough surface kind of helps for the bone cells to integrate and have a good sort of bond between the implant and the bone. There's a lot of chemical treatment which is done. Chemical treatment as such is not like different types of chemical, but they use hydroxyapatite, which is one of the most important content of the bone. And so hydroxyapatite has been very commonly used. So here are some examples where you can take hydroxyapatite and use plasma to put hydroxyapatite on these implant surfaces and texturize at the same time. So you have hydroxyapatite, sort of a coating and a texture which is there. A lot of these textures have all been at a micro scale, and our work has been mostly at a nanoscale. And I'll explain why I think nanoscale is important. I'm not saying microscale is not. We definitely need a microscale topography on a surface, but we also actually need a nanoscale topography, because if you start looking at bone, and if you start sort of dissecting bone into its different components, what you find out is that bone is made up of nanomaterials. Bone is made up of collagen. Bone is made up of hydroxyapatite. There's a lot of different types of proteins which are there. Structurally also, there is fibrous kind of a structure. There are micropores, there are nanopores. So at a very core level, if you start looking at the structure of the bone, you do have a lot of nano features along with micro features. And so the idea is that if you can now create material surfaces on implants, which have this micro nano topography, along with whatever biochemical, along with all the biochemical properties that you need, then you should be able to see a good interaction of the bone cells with this implant surface. And when these implants are actually put inside the body, the bone cells can migrate to the implant surface, grow new bone, form a strong bond, and in that way, this implant is fixed. And hopefully it's fixed such that you don't have any loosening of the implant that's going to happen ever in future. The idea is that we want to actually have these implants last forever. We don't want to deal with revision surgeries, because every time you do revision surgery, it gets more and more complicated. So that's what the idea is. And that's what kind of prompted us to go more onto the nano side. Now, I'm going to talk about this very simple method that we actually use in my lab. And over the years, we have developed many different types of methods, but this is something that we have used quite a bit. We actually try to make these nanoporous surfaces. I'm going to show the SEM images even before I talk about the process and everything. So essentially, this is a process. It's called anodization. Anodization is a very common process, which is actually used to make nonstick pans. It's used in many other things, but nonstick pan is something that it's very easy for us to visualize. A lot of these are aluminum pots and pans, and they do this hard anodization. It's a very traditional material science technique, material engineering technique, where you dip it into a hot acid very quickly and take it out, and it actually hardens the surface and also creates this non-stick coating on the surface. It's essentially the same process, but it's not a hard anodization. This is a soft sort of anodization. We don't put it in a hot acid. We do this in a slow process where we have titanium as anode. We use a cathode. In our case, we use platinum as a cathode. We have an electrochemical setup. We connect to a power source. And now by changing the amount of voltage that you apply, you end up with surfaces that would have different size of these nanotubes. Now, these are nanotubes which are also made from titanium. This is not something that's deposited on the surface. You have your material and you're reacting and you're using the bulk of the material to form these nanotubes. And if you look at naked eye, it looks the same. The surface doesn't look any different. But when you look in SEM, you see that you have this very interesting topography there. And the diameter increases as you're actually changing the voltage. And the electrolyte that we use here is hydrofluoric acid, which is actually pretty dangerous acid to work with. But if you use proper precautions, it's fine. But if you change that electrolyte, then you actually end up with a surface that looks like this. So you have a surface like this or a surface like this. So touching tubes, not touching tubes, you can control the gap, you can do many different things. These are perpendicular from the surface. So these are actually perpendicular from the surface, which are growing. And so these surfaces we have used quite a bit. The interesting thing about the surface is that they're actually extremely hydrophilic. So I'm sure everyone knows what is hydrophilic, but what I want to show you guys here that this surface is there less than 10 degrees. So we actually classify them as a super hydrophilic surfaces. Super hydrophilic means extremely high surface energy, which means that if you have proteins and everything that come on the surface, they very easily can spread. The cells can also very easily spread on the surface, which is actually very important for orthopedic applications, for the cells to be able to spread on the surface. We do a lot of characterization. I'm not going to spend a lot of time here in characterization today. We do XRD and XPS and a lot of different, surface charge and corrosion measurements and all these things for the surfaces. I won't talk a lot about that because I want to kind of talk about the, from a biological perspective, why these are so exciting. So we have done a lot of studies. Here I'm going to talk about studies that we have done with adipose derived stem cells Adipose-derived stem cells are mesenchymal stem cells, but they are from the fat tissue. We used to use mesenchymal stem cells. We have used mesenchymal stem cells for many years. We were actually isolating MSCs from rat bone marrow. We have also done some human work, human MSC work. Eventually, we decided to go to ADSCs because ADSCs, they are sort of easier to get it. And there's a lot of adipose tissue which is thrown away. Doctors, when they do liposuction surgeries and when they remove fat tissue, that's all thrown away. That's rich of stem cells. And these stem cells have the same potential as the one from bone marrow. So we have been using these adipose-derived stem cells. And so basically what we do is we take these stem cells, we actually put the stem cells on our material surfaces and allow the cells to grow. We want to see that how are the cells growing or not. We provide all the appropriate conditions and what is needed. And so here is one of the examples. So I don't know if you can see on your screen, but there's a lot of blue dots also which are here, and the blue dots are the nuclei, the nucleus of the cells, and the red is the cytoskeleton. And you can see that compared to titanium, we do see more cells, and I think the topography is providing those cues for the cells to actually attach better and to grow better and to divide better. So we eventually end up having more cells on these nanoscale surfaces compared to a surface which didn't have any sort of topography. Just to give you an idea, this is the SCM image of the cells, and you can see that these are cells, the dark spots, but you can see that by 7 days, this surface which had nanotopography was completely covered. And that's because of the surface, because it's a superhydrophilic surface. At the same time, you have the topography, which has a size, sort of similar size range that of the microscale, the nanoscale features of the actual bone, which is there. For bone cells to be able to start depositing bone, it's really important for them to start forming these clusters. So these cells will first attach, they will then divide, and they would form these clusters. And these clusters of few hundred cells would start depositing bone, which is basically depositing hydroxyapatite, which is basically depositing calcium and phosphorus and converting into hydroxyapatite on the surface. So we have done a lot of studies with that as well. Here is just a study that I've just kind of given an example. So we can take these adipose-derived stem cells, we can grow them, and then we can kind of give them signals so that they become bone cells, and then we see how they're depositing the bone, proteins and things like that. So here is another, and again, as I said, I'm just giving like example results. We have a lot of characterization, but I just want to kind of pass the message that, nanostructured surfaces do influence cells. And it's not specific to, and it's specific to a cell type. It's not that all the cells will do that. There are some cells which will do and some cells which will not do it. And in our case, this adipose-derived stem cells, the surfaces, they support the cell growth and the differentiation, which is shown here. This is osteocalcin, which is a protein which is expressed at a late stage. As the cells are now making the bone, they're also going to produce this osteocalcin. And this osteocalcin is sort of a marker that the cells are healthy and they are actually doing what they're supposed to do. So all the green dots that you see is osteocalcin, and we see way more osteocalcin deposition on these nanosurfaces, after 3 weeks, which means that the cells are becoming bone cells and they are growing. They're depositing new bone. Now, translating this in vivo, how would this work? Well, in vivo, if this is happening, then we would start seeing bone deposition, which means the implant would start integrating with the natural tissue, and that's what we want. We want this to happen faster. We also want this to happen in a way so that the bone deposition is going to be stronger and you don't have any loosening of the implant that's actually going to happen. Okay, so I'm going to talk about cardiovascular applications as well. As I said, I'm very happy to have discussions, with anyone who wants to talk more about, this, separately, just to follow the time today. I want to kind of cover these two topics. But anyone who wants to talk to me, feel free to send me an email. And since I'm already here in India, it's much easier to talk and don't have to worry about time difference and things like that. So one of the other focus that we have in the lab is on cardiovascular disease, and specifically on cardiovascular implants. We work on mechanical heart valves. So mechanical heart valves, it's a titanium casing, and then you have 2 leaflets, which are actually made up of pyrolytic carbon. I'm not going to talk about that today. We have done a lot of work on making these leaflets anti-clotting, because one of the problems with mechanical heart valve is as the blood is flowing, the hinges and everything, you have a lot of blood clot that happens. And over time, the heart valve is not able to open, very freely. And then, people would have problem. And the other implant that we have worked with is Stent, which is what I'm going to talk about today. But cardiovascular disease is, as you all know, it's a leading cause of death in the world. And it costs a lot of money to mitigate this disease. So people who have this disease, they spend a lot of money, medications and surgeries and things like that. And in general, when we talk about cardiovascular disease, we are talking about disease that affects heart and blood vessels. So it's not always about heart, it's about blood vessels. And blood vessels are in any part of the body. So people could have clotting in the blood vessel in the legs, and that's bad too, because that's going to affect heart eventually, because it's all connected. So typically, as you can see here, and I always try to talk about this, and I could not find something similar in India. I'm sure that there is data like this in India as well. Maybe I didn't know the right place to find it, but this is for United States. And this is something which kind of shows geographically as well, how it's distributed. It also depends a lot on the parts of the country where what kind of food people are eating and what kind of diet people have. But, in United States, people, every 40 seconds, somebody has a heart attack. And that's really bad. And a lot of this is related to the cultural aspect of where you live and what kind of food you eat. But at the same time, educating people, how to prevent heart disease from happening is a very important sort of outreach, that needs to be done as well. But when people have heart disease and people have cardiovascular disease, they have to get it treated because it is life-threatening. And there are different types of disease that can happen. So here is one example of blood vessel disease, which is what I'm going to talk about today. It's atherosclerosis, which is basically plaque that builds up into the blood vessels. So you have these blood vessels which are carrying blood, they're carrying oxygenated blood, oxygen-rich blood to the different parts of the body. And you can have a plaque that actually builds up, and that can be for many reasons. It could be fat, cholesterol, calcium, many times genetic reasons also as well. Many times the cultural aspect of how you eat the food and what you're used to eating, that can also result into this plaque formation. So what happens is, as the blood is flowing, this plaque is going to be there, and this plaque is going to obstruct the flow of the blood, which means that you'll have limited blood that's going to reach different parts of the body, which means limited oxygen that reaches, and then it can cause a lot of problems. It can cause heart attack, stroke, it can be deadly. And it's not just that it affects heart or blood vessels near heart. This can happen in brain and in arms, in legs, in kidneys. It can happen in many different parts of the body. How do we treat this? Well, there are two ways you can treat this. One is basically a bypass. Well, bypass is not the first option normally. They always try to do this, percutaneous coronary intervention, where they actually open the blocked artery. Sometimes they inject a balloon and let that open up, or sometimes they put a stent, which is a mesh tube, and it keeps that open, and so the blood can flow. If that doesn't work, or if for many people, if there is no chance of doing this stent, then they will do the bypass. And bypass can be actually pretty severe, pretty major surgery, because you're literally stopping the heart and the lung and connecting the person to a heart-lung machine, and then doing the repair what that needs to be repaired. And when you do bypass, you can actually use allografts, or you can use autograft. You can use allografts, or you can use synthetic grafts. So synthetic grafts will be made from Teflon, Dacron, materials like that. I'm going to talk mostly about stents today. And there are two types of stents which are currently in market right now. You have a bare metal stent, which basically is just a bare metal. It's just a mesh-like structure, a bare metal, or you have a drug-eluting stent. Now, both of these have advantages and disadvantages. And the next slide, is going to talk about what is that. So when you have a bare metal stent, it's just a mesh tube. You put it in using a minimally invasive process. You put that in, and then the stent is opening, and then it allows the blood to flow. But one of the things that happens with this bare metal stent is something called restenosis, which is shown here in this figure here. When you put this stent, it's going to now denudge some of the endothelial cells. You have endothelial cell lining in the inside lining of the blood vessels. When these endothelial cells are denudged, means they're actually removed. Below it, there is smooth muscle cells. These smooth muscle cell layer gets exposed, and these smooth muscle cells then start dividing. And once they start dividing, they would cause this restenosis, which is happening. So the whole reason you put the stent, the same thing is happening, not because of plaque, but because of restenosis. And this can happen in many people. Now, it's very hard to predict it's going to happen for a certain kind of a patient, but it will happen. To a certain number of people, it may not happen to a certain number of people. To prevent this, they came up with this drug-eluting stent. The drug-eluting stent is the one where they take the stent and they coat the stent with a drug that actually prevents this division of the cells. So it does prevent restenosis. But once that drug comes off, because the drug is coated in a polymer coating, once that comes off, you have thrombosis that happens, which means blood clotting is going to happen. And it's a late thrombosis. So it doesn't happen right away. It can happen after a few months or 6 months or 8 months, where you would have blood clotting that happens, which is also, again, a problem. Blood clotting is even a bigger problem, because if that clot detaches, and if it's just free flowing in the blood, it can reach brain, cause stroke, it can reach different kidney, cause kidney failure. There's a lot of different things, bad things that can happen when you have a blood clot inside the body. And so restenosis and thrombosis are sort of the key reasons why stents fail. Now, the interesting thing is that this doesn't happen in everyone. There are people who have stent and they live with the stent for the rest of their life. And there are people within 3 months, the stent fails. It's very hard to actually predict what's going to happen. So our approach has been that, can we now come up with some sort of a surface that would prevent blood clotting from happening? And to be able to do that, we need to understand, first of all, how blood interacts with the material surface and what are the different steps that happen in blood clotting. So, there are two sort of cascades here in blood clotting. So you have basically a thrombosis cascade, or you have an inflammation cascade. Thrombosis typically is where you have protein that absorb, platelets attach, activate, red blood cells get trapped, and then eventually have blood clotting. Inflammation is where, again, you still have protein absorption, but you do have inflammatory cells, and then they have foreign body giant cells which are formed, and then you have this fibrous encapsulation. This typically does not happen a lot with cardiovascular implants, but it could happen with other implants which come in contact with blood. And then the third part here is this vascular injury, which is basically now the endothelial cells are damaged and the smooth muscle cells underneath are exposed, and that's why they're dividing. So that we have to understand from a materials perspective how each and every step is happening and what we need to actually control so that the progression of this process does not keep on going on and it's stopped. So our approach, again, has been to use this titanium nanotube arrays as a surface for stents. And I'll tell you the reason why just in a minute. But the idea is that you create these nanotubes and maybe eventually fill these nanotubes with drug. This is not a polymer-based drug delivery system. You don't have any polymers here. It just filled into these tubes. So you don't have to worry about polymer degrading and then, you are going to have any thrombosis because of that reason. But the reason why we think these nanotubes would work really well, because if you look at the architecture, the surface architecture of the inside of the blood vessel, it looks like this. It's called endothelial glycophilics. It's this hair-like structure which is there. It's a flexible structure. But it is a hair-like structure. There's a nanotopography to it. And our idea is that if you can replicate that nanotopography through these nanotubes, even though these nanotubes are not flexible, they're rigid, at least we are actually able to mimic that structural aspect. We might actually see some advantage in terms of, preventing blood clotting and things like that. And so once we had that hypothesis developed, we did a lot of work with blood, and we basically go to the blood bank on campus, and students find volunteers to take the blood, and from the blood we remove the platelet-rich plasma, and then we put the platelet-rich plasma on the surface to see how the cells are actually attaching. So you can see this titanium and nanotubes, and this is actually very interesting. So on titanium we see a lot of platelets. The big cells here, the fuzzy-looking cell is a leukocyte, white blood cell. Whereas with nanotubes, we don't see the clusters form. So here, the superhydrophilic surface is now actually preventing the clusters from formation because the cells are very smaller. Platelets are much smaller than adipose. If you remember the adipose derived images, there was a lot of spreading. Here we don't. So we think that the surface topography is now preventing the cells from actually forming these clusters, which is the precursor for blood clotting to happen. We also look at hemolysis, which is one of the other important things, because if your surface is going to now kill the red blood cells, it's bad. So we do hemolysis as well, where we try to see how the red blood cells are lysing and how they're releasing and what we find out that the hemolysis is also less on these nano surface. We also kind of got interested in looking at some foreign body giant cell response. So again, we can take the blood, we can remove the plasma, we can remove the white blood cells. And then once we have these white blood cells or monocytes, we can put the monocytes on the surface and kind of see the progression of these monocytes as it's shown here. This is like a model system where you have monocytes that here, they become macrophages, they form fusion, and then they form these foreign body giant cells, which is basically, at which the inflammation, is going to happen. So we have done some of those studies, and as you can see here, that we can prevent that as well. We cannot eliminate it, but we can prevent that as well on these nanotube surface, where we can reduce the formation of these foreign body giant cells. So this kind of got us thinking that, yes, we do have the topography which we needed for these stents, but we don't have the chemistry, because endothelial vacuum calyx, yes, it's flexible. It's hard to make something flexible on a metal surface without actually putting a coating, because we don't want to put a coating. We have the same problem like drug-eluting stents. We want to use the bulk and grow something, which is why we use nanotubes. So we wanted to see that maybe we can do some type of chemistries which are not going to be traditional coatings, where you have the problem of coating degrading, but attaching smaller molecules. So one of the things that we have done is use polyelectrolyte multilayers, and we have formed different multilayers on these nanotube surfaces to see that if that can give the chemistry or the biochemical properties which we actually needed. So we have used different polycations, polyanions. We have used chitosan, Tanfloc, heparin, hyaluron, different combinations to make these polyelectrolyte multilayers. This is just to give you an idea about, the chemistry of these. The main goal for us was not to compromise the topography. So as you can see here, the top one here, when we do these 5 layers of polyanion, cation, anion, cation, 5 layers was still able to preserve the topography, whereas 19 layers was not. So we never bothered to do 19 layers. We always went with 5 layers, because for us, it's important to have that topography and the chemistry combination. And then when we did some studies with platelet-rich plasma, what we found out was that the Tanfloc heparin was actually pretty good. If you compare these to the nanotube picture that I've shown a few slides back, this is even less than that. So we are getting a little bit better in terms of preventing clotting on the surface. Some of these were even worse, so chitosan heparin and chitosan hyaluron were worse, whereas Tanfloc heparin was actually the best one. And this is the nanotube control here that was also shown in the previous slide. The other approach that we have used, so again, I'm talking about these different ways in which we want to try to prevent blood clotting on these material surfaces. We also use nitric oxide. Nitric oxide is something that is naturally released in our blood vessels through the endothelial vacuum calyx, and it's known to be a potent vasodilator and also inhibits platelet activation. So we wanted to develop some chemistries on the surface using these nanotubes, using polyelectrolyte multilayers, so we can release this nitric oxide. And this nitric oxide would then prevent platelets from, attaching and activating on the surface. Again, as I said, the goal is to make sure that the topography is maintained, which we did. So even the nitric oxide didn't really damage, putting that didn't really affect the topography. We can see the flux of the release, so how that nitric oxide is released. And nitric oxide is a very small molecule, has a very short half-life. So you want to release it, and you want that to be recognized by the cells right away. If it remains for a longer time, it gets deactivated. So we were looking at short release times. And as you can see here, in 20 minutes, we get quite a bit of nitric oxide release, which probably is good enough for the platelets to not get activated and not start this whole clotting cascade, because, clot happens very quickly on a surface. So when we did some of the work with platelet-rich plasma, what we found out here is that the nitric oxide was even further able to reduce the platelet activation and adhesion. So as you can see, we are looking at the synergistic effect here. Topography, we know it works. We know PEMs work. Now PEMs with nitric oxide, also it actually works. So the idea is that we want to create surfaces on which you can prevent clotting, so that the stent surface can remain free, and we don't have to worry about, removing the stent, doing bypass, and all these different kind of things. So these are some of the fluorescence images, some of the SCM images. You can see that, this platelets, they are flat, and you see some of the extensions coming. Those are activated platelets. This is a platelet aggregation that happens. We do not want platelet aggregation. We do not want platelet activation. We do want platelets. Platelets are important, but we don't want them to activate on the surface. And you can see with the PEMs and nitric oxide, you have the platelets, You have fewer platelets, but they're not really activated on the surface. And that's what we want to do. I know we just have a few more minutes, so I'm going to try to wind up. We also have developed a lot of superhydrophobic surfaces for blood clotting applications. I'm sure everyone has heard about superhydrophobicity. Lotus leaf is the best example of a superhydrophobic surface. And the reason why lotus leaf is superhydrophobic because it has a topography, but also has a wax-like coating on it. So combination of both of this created a superhydrophobic coating where the droplet just slides from the surface. So our approach has been, can we use topography, the nanotube topography, but can we maybe then put something like a carbon fluorine bond on it, which would make it a superhydrophobic? So that's what our sort of hypothesis for this work was. And the idea of superhydrophobicity, and again, I won't go into the detail of it because we want Cassie-Baxter state. Basically, we have this texture, and if we modify the texture with the chemistry, when you put a drop of liquid, it remains a drop of liquid because there is air that actually gets trapped. Very small amount of air that gets actually trapped. And that's why you have this rolling effect, which is there. You can see some of these pictures here. So we have blood, we have the platelet-rich plasma, and we have water on a superhydrophobic surface, super repellent surface, and they remain as a drop. They don't spread. If they don't spread, there is no chance for proteins to absorb. There is no chance for cells to be in contact. And hopefully we won't have any clotting that would happen. But we wanted to kind of show that this is what's going to happen. So just to give you an idea, again, this kind of modification, we can still preserve the topography. The contact angles are pretty high, more than 150 degrees. The nanotubes by themselves, they are super hydrophilic or hydrophilic. But once you start putting these, fluorosilane or chlorosilane, you can get superhydrophobicity. You can get contact angles which have minimal contact with the surface. And I'm going to show some videos here, kind of give you an idea about what do I mean. So here is a video, and it's going to replay again and again, I think. So this is titanium surface, just a drop of water and how that's interacting with it. Let's look at a titanium surface, which we modified with that silane, and it's a hydrophobic surface. Well, sort of hydrophobic, or almost hydrophobic surface. Nanotubes, they are super hydrophilic. So you can see that the water completely spreads on it. But look at this one here, which is the nanotube with that silane, which is super hydrophobic. It's a slow motion video because that's the interesting thing to actually see. So see, the water droplet doesn't even want to stick on the surface because it's a super hydrophobic surface. It just bounces off. So imagine that now blood was flowing. Blood will flow, but there would be no contact of the blood with the surface, which means nothing can attach on the surface, which means we can prevent clotting happening from the surface. So we have done a lot of sort of mechanistic studies with this super hydrophobic surface. We have done like protein absorption. Again, we find that there is significantly less protein absorption on on fluorosilane, but not on chlorosilane, even though both are hydrophobic. We have also done platelet studies. You can see that the fluorocarbon has quite less, not as less as nitric oxide, but nitric oxide is way more complicated chemistry than this one. And we see quite a bit less activation of the cells as well. Here are some videos that, I'm almost about to end because I know I want to leave a few minutes for questions as well. But this is now real blood. So you can see the real blood on a titanium surface, and you can see how the blood is spreading. And now the student, he's trying to lift to kind of see that, when a flow happens, and we have done flow studies, I'm not showing that here, but you would actually have a streak that's formed, and you'll have blood that's going to stick on the surface. If it's flowing, you'll have a lot of blood that sticks, and more blood that sticks. You'll eventually basically have clotting that's formed. Let's look at the nanotube. It's a super hydrophilic surface. You would see that when you put the blood, it is going to spread. It spreads even more than it spreads on titanium, which is what's expected. And when it's lifted, you would see that a lot of the blood can come out, but still you're going to have some clotting, which is going to be there. Now, this is a super hydrophobic, which is the chlorosilane, not a fluorosilane. You can see the drop of blood remains a drop. And I'm a student, he's going to try to lift it. And you can see that the drops just slide out, and you don't have anything that's remaining on the surface. Just a little bit slower video, but now you can see that most of the blood is gone out. And this is the fluorosilane, which was the best one, where you can see that the drop of the blood doesn't even want to stay, didn't even need to lift the surface, it just slided. just slightly, because the surface had such a low surface energy. And so surface like this would be really, really good for preventing clotting. And that's what our work, currently has been. We have also done like whole blood clotting to understand the clotting kinetics here. So what we do is we take materials, we put a drop of blood, we allow the blood to clot, then we put water, and then we kind of remove the samples and dissolve all the blood. And you can see that more red means that there is less clotting, because the blood, and less red means more clotting. The reason why, because when the clot is formed, there's red blood cells which are free to lyse, and so it's opposite. More red means there is less clotting. Less red means there is more clotting, because a lot of the red blood cells are actually trapped. And that's what we find out, that mostly with the superhydrophobic one, we probably see very minimal clotting that's happening. Whereas with the non-modified ones, we see that there's clotting because there's drastic drop in the hemoglobin that's happening on the surface. So some of these are ongoing studies. In my lab, we are still continuing to work on this. We are using some new types of surfaces. We are using some hydrothermally treated surface. Some of these are not published, so I have not included here yet. But we are using some other types of methods as well to create these texture nanotopography on the surfaces, and trying to understand, what is sort of that optimum, surface topography that we can then now modify with nitric oxide or with, PMs or things like that, so that we have the best non-clotting properties. The key of this is that nothing attaches on it, which is also bad, because for stents, we do want endothelialization to happen. And so that's the other thing that we are trying to now kind of figure it out, that yes, when the blood flows, nothing is going to stick on it, but there are cells around. We want the cells to actually grow on the stent and stent to get integrated. That's the next task, that we have now to figure out, that how we can now prevent clotting, but improve endothelialization of these stents. So I normally don't have, I just have sort of like a conclusion slide, like a summary slide, just kind of summarizing, but, just to point out that a lot of our work is in materials engineering. We engineer materials. We don't make new materials. We take existing materials, but we want to engineer these materials even better, so that once they come in contact with the biological system, those interactions are in a well-controlled way. And then in that way, you can now kind of, eliminate some of the problems which are there because of this mismatch between the material surface and the tissue. We use nanotechnology as a tool. It's never the answer for us. That's what we found out. It's not the answer. It's a tool that actually enables us to do things in a much more effective way, much better way. And so we kind of lie into this intersection of this Venn diagram here, where we work with materials, we work with biological interactions, and we use some of the nano sort of technology, not nanoscience as much, but more sort of the nanotechnology approach to come up with surfaces that if they're part of these implants, and these implants, when they're put inside the body, we won't have any sort of rejection that would happen. So with this, thank you very much, for listening to my talk. I know that I was hoping to finish 5 minutes earlier, but I'm sorry if I took more time, but I think we still have a few more minutes for any questions.
Yeah.
So thank you. Thank you very much.
Yeah. First of all, really, thanks a lot, Ketul. Really, very, very fascinating talk. Before we take questions from the colleagues, just a small announcement that this was the first inaugural talk for the Science for Society lecture series. So we had students from IIT Gandhinagar, Naipur, Ahmedabad, Indian Institute of Public Health, GSBTM, Guj Coast, GBRC, Ahmedabad University, Nirma University, and Gujarat University. So I think around 10 institutes students. So I'm sure this talk must have triggered some more exciting ideas within your minds, because we know that in this area, a lot of faculties and students are doing some bits and pieces of research in different aspects. And I'm sure this talk by Professor Ketul must have acted as inspiration for you all. And before we take questions, a small announcement that Ketul is here in India. So we're trying to get him here to IIT. maybe we'll send a small announcement. We can have lunch together on a Saturday or something. So we'll make that arrangement and we'll get back. So now the floor is open. We have a couple of minutes to discuss any questions. So feel free to switch on the mic or put in the message box with any questions that you have.
Thank you for inviting me as the inaugural speaker. It's an honor for me as well.
Yeah, thanks. Yeah. So as we say, like, make local as vocal. So you are local, and
yeah,
we have a right to invite you.
Yeah. And
maybe I can begin.
Yeah.
I can begin with so this is the first part, which you said the titanium based coating for this hip bone. What as we all know that with time these materials get degraded, as you showed. Can we make it like in the contrast? Like we have different packets of the material. Like let's say when there are some sensors that is the first layer of the material gets degraded, the second layer comes up or third layer comes up, and by this, if you like kind of enhance the lifetime of these materials.
No, certainly we have. So that's some of the things that we are doing right now is we are trying to like combine hydrothermal processing along with this anodization to actually create. So when we think about layers, we are thinking about more like hierarchy, So we're thinking more of like, can we have nano as sort of the first sort of surface that the cells see, but then eventually we have some sort of micro scale topography that long term, helps in better integration. But yeah, certainly I think we have looked at that. I think with the stents, this is going to be more critical,
Right.
Because with orthopedic implants, once you put it and once it attaches, it's all done. Whereas stent, it's always going to be exposed, and we are trying to kind of figure out like what in long term would work the best, because eventually if proteins attach, the topography is going to be lost. So we want to use like a micro nano kind of a topography so that we still have some micro scale topography eventually that would help with the prevention.
Absolutely. Yeah. So Dhwani has a question. Thank you for the interesting talk. For the cell culture assays, how do you fit the titanium based nanotube arrays inside the 96?
So we normally don't use 96 because we normally like to use 48. So, when I was working many years back, when I was a postdoc and a student, we always used to use like a 24 well plate. When I became a professor, I'm like, well, that's a lot of media and a lot of stains. So we moved to 48 well plate. But typically our samples are about 8 millimeters disc when we use discs. Or when we use some square pieces or something, it's like 8 millimeters by 8 millimeters or maximum 1 centimeter by 1 centimeter. And which, sometimes we comfortably can put it in a 48 well plate. But we do 96 well plate for all the assays. So all the assays are 96 well plate. Yeah. That's a good practical question. Nobody asks those kind of questions. So thanks for asking that.
It's a good audience. Like we have that thesis.
Yeah.
We ask practical questions.
Yeah.
So other thing, what I was just thinking is in the same lines, that is nanotube arrays which are putting. One idea would be to make them more compatible is first coat them, maybe say with the extracellular matrix, which is derived from the stem cells itself. Because then, when you're putting it on the cells or tissues, they don't see the foreign material. But I guess that would be like cost.
No, so I think
like
one of the things that we have always kind of thought. So the media kind of replicates some of the things that we have in the ECM. So when we put the cells with the media has all the proteins and everything. So one of the other things that the thought process behind this experiment was that we want to try to make surfaces such that how the implants will be. So if you have an implant, with cardiovascular implants, they dip it into like clotted blood to prevent clotting. That's what they do. Like sometimes they put the stent in the blood and then they put it so that it's already pre-clotted, so further clotting cannot happen. With orthopedics, they typically just take the implant and put it. There's not a lot of conditioning that happens. So that was sort of our thought process, that we want to not put anything on the surface. Let's see if the cells deposit their ECM. And is that surface promoting them to do the ECM deposition or not?
Perfect. Yeah. That seems a really good.
Yeah.
Any other questions, colleagues? Feel free to switch on your mic. Yeah, Carla.
Hi. Hi. Good morning, Dr. Ketul.
Good to see you, Carla.
Good to see you too. Yeah, thanks for a nice talk. So my question is with regards to the nitric oxide eluting stents. So how do you make sure that you deliver the optimum concentration of nitric oxide to prevent cellular toxicity?
That's the golden question. We don't know if it's optimum yet, because we haven't done a lot of in vivo work yet. The hard part about nitric oxide has always been making sure that, so right now, I think we're delivering more than what it should be. I think we're delivering more, and more nitric oxide is also toxic, So I think we are going to do some in vivo studies in this year. We were going to do it last year. We didn't end up doing it, but we are planning to do it. And again, with the in vivo studies, we are not really sort of optimizing nitric oxide. We are just using 3 different fluxes to see what gives us the best result and kind of figure it out that way. But yeah, that's the hard part, because we don't know if this is optimum or not. I think this is more than what it should be. Just to be on the safe side, we are putting more nitric oxide donors so that we can see some changes on the surface. But yeah, there's a really small amount in the blood released by the endothelial cells. And that, I think, is probably going to be very hard to achieve with the chemistry that we are doing, because if we can, then we won't be able to detect it. That's the other problem. But yeah, I think you are right on that. It's not optimum. I think we will do some in vivo studies. We'll have a better idea about, if we find out that 3 fluxes we use, and if the lowest one is still the same as the highest one, then we know that it's still lower than that.
Okay, thank you. Good luck on that.
Thanks. Yeah, just in connection with that, Ketul, I'm sure your team must have looked at the immune responses, most of these times when you put this foreign materials, lot of cytokines are secreted, or a word, maybe I didn't see that in the slides, but I'm sure you must have done.
Oh no, we have done it. This was almost about 7-8 years back. We did the whole cytokine profile, TNF alpha and IL-6, all these cytokine profile.
Yeah.
One of the biggest worry with nanoscale materials is always the immune response. It's always or inflammatory response. Is that the one that's going to be the preventing in using? And we have done it, and what we have found out is that in many instances, many of these cytokines, they were lower than what the control is, or the same as control. So I think we are not too much concerned right now. And also because we think about this as surfaces and not nano, because if we had nanoparticles or something, then there's whole different issues, with free floating nanostructures, whereas these are sort of attached on the surface. We didn't really see, we didn't really find that this is going to get even worse. Actually, it might be even better in terms of some of the immune response.
Absolutely, yeah. Okay. Any quick questions? We have maybe a couple of minutes. Students, especially.
And if anybody has questions later on, feel free to, it's easy to find my email address on the web. You just type my name and you'll find out. And you can always email me if you have any questions. And I'm happy to talk to anybody. I'm here for another, 6-7 weeks. So I can, even when I'm not here, even when I'm in US, I don't have, it's absolutely fine to talk to me. It just, we'll have to figure out a appropriate time.
No, we'll get you here. Yeah. We'll have lunch. Yeah.
Yes, sir. Good afternoon, sir.
Yes.
Yes, sir. Sir, can we not replace the chemically synthesized nanomaterials with the biologically present bio-nanomaterials?
So, yes, I think there is a possibility to do that. Many years back, we tried doing that. We tried doing some nano texturing with collagen, use collagen and do that. And we then eventually didn't end up pursuing it. But yeah, certainly, I think you can use biologically active nanostructured surfaces. So surfaces which have nanostructure, which is made from a biological material rather than a synthetic one like this. It also depends a lot on the application part of it. For example, with stents, they do have now polymeric stents. They are now, people are working on polymeric stents, but one of the big problem is, it collapses. So one of the problems with biological material is that you may not have the strength that you need that this nickel titanium or titanium based stents actually give. And the same is also for orthopedic implants. With orthopedic implants, there has been a lot of work putting biological, biomolecules on it and coatings. We were trying to go away from coatings because, when you put a coating, it's actually on top. and you have problem with delamination, you're putting something on top. You wanted to use a process which use the bulk and, like a bottom-up process rather than a top-down process. But for many applications, you could actually use biologically derived material and then make textures or surfaces out of those materials.
Yes, sir. And sir, so is degradability the readily encountered disadvantage of bio-nanomaterials compared to chemically synthesized nanomaterials?
Not so much. When I think about degradability, I always think about, like, for example, with stents, they are trying to make degradable stents. And I would say that maybe that's not the best option, because the disease, a lot of, doctors and everyone has known that, when people have heart disease, it comes back. And if you have a degradable stent and if it goes away, then you might have chance. So it's better to have something permanent. But certainly, I think it very much depends on, like, for example, if a piece of bone is lost, like a chunk of bone is lost, something like that, you definitely want a degradable material. Something that you want a scaffold which has micro nanoporous pores or things like that. But when we are looking at a whole implant, I think making a degradable implant would not be the best option, because for that application, we do need we need the stability and we need the mechanical support. And that's why we want to use metallic based kind of implants. I hope that makes sense.
Yes, sir. Thank you so much, sir.
Yeah.
Okay. Not much questions. I think thanks a lot, Ketul. We really enjoyed your talk. It was really very inspiring. I'm sure many students will write back to you for sure for more discussions, and because we faculties will definitely touch base with you for some positive. Like, we are doing some very good work, similar work here at IIT. Maybe like when you visit us, we'll discuss more on the specific projects and joint ideas. I'm sure DSB team will be equally excited on these projects. So thanks a lot, and let's plan a small meet.
Definitely. And thank you everyone for coming, and thanks for inviting me.
Thank you everybody, and on behalf of GSBTM and IIT Gandhinagar, we thank you all for this talk, and with this we close the session. Thanks everyone.
Thank you. Bye.